You're most welcome to this talk. Thank you for coming back. Now, there's a lot of vaccination per capita going on in Israel. It's all the Pfizer vaccine, and they are following the phase 3 clinical trial of giving one dose, the primer dose, followed 3 weeks later by the booster dose. So the data we've all got is based on that, but there is a lot of really good data, and it has to be said, very promising data coming out now. Now, this is the paper I want to review here. It's from the New England Journal of Medicine. It's peer-reviewed. It's there for the world to see. So let's just focus on that a bit and work out what it's saying, because it is quite hard to understand. It takes a while to read through it to sort of work it out, really. But let's get on with it. So this is the Pfizer vaccine. Messenger RNA vaccine, COVID-19, in a nationwide mass vaccination setting. So again, the real world data now is starting to come in. And this is just published a few days ago, 24th, I think this was published. Now, the method here, so they took vaccinations that were done between the 20th of December 2020, and this data set goes up to the 1st of February 2021. So we've got all of January's data there and the end part of December's data. So it's a fair chunk of data. And the other thing about Israel at this time was it's kind of ideal for research, really, because they were rolling out the vaccine, but there were still a lot of cases. The prevalence and the incidence in Israel was still high. So there was quite a lot of sort of things to compare it to, really. So it's a bit of an ideal situation from a research point of view. Now, what they did was they took people that were vaccinated and they matched them to unvaccinated controls in a 1-to-1 ratio. So what they did was they took people from Israel who had been vaccinated and then they matched them. So similar ethnicity profile, same sex profile, same to some extent same comorbidity profile. But what they didn't manage to do very effectively was match them for age. Now, the reason for this, and this is, you could say this is a major flaw in this paper, but it's actually not, because we can account for it quite easily. Because obviously they were vaccinating the older, more vulnerable people first. So the people that were being vaccinated were in the older age group, and that means that the controls, the people they picked to sort of compare and contrast to form a control group, almost by definition had to be younger. So that means any effect of the vaccine in this older age group, any efficacy that was seen, is the lower estimate. The lower estimate, because we know that older people are more prone to severe illness, hospitalisation and indeed death from COVID-19. So actually what it means is all the results in this paper that show good efficacy are minimum effects, because they were comparing older people that had been vaccinated with younger people who hadn't been vaccinated, and those younger people are less prone to more severe disease. Hope that makes sense. So that's the weakness in this. The controls were younger, but as I say, we can account for it, and it means that it's minimum data. So according to demographic and clinical characteristics, so they matched. Now, they ended up with a number in this particular cohort at 569,681. That was the number that had been vaccinated. So they picked a control group of younger people who were exactly the same number. So in other words, this is getting on for 1.2 million study. Now, from memory, the Pfizer phase 3 clinical trial study, I think from memory, the number in that, it was a good size study, but it was just under 28,000, the original phase 3 clinical trial for Pfizer. So this has gone up from a clinical trial of just under 28,000 to 1.2 million, as indeed it was 1.2 million coincidentally with the Scottish data we looked at a couple of days ago. So very impressive data sets now being recruited, as we would expect, because this is post-release information. Now, documented infections. So we've got this group of 1.2 million people. We've got this time span here from the 20th of December to the 1st of February. And amongst that 1.2 million people, in that time, there were 10,561 documented infections. So they had that many infections, that many people. Half of those had been vaccinated, half hadn't been vaccinated. So we've got the vaccination group and the control group, and I can think you really can see this is a very rich potential data set, as indeed it is. Now, of this 10,500 basically, 5,996 had symptomatic infection, so 57% were symptomatic infection. Remember, half vaccinated, half not. Requiring hospitalisation, well, of these 10,500 infections, 369 were poorly enough to require hospitalisation. Severe cases were classified as 229. So what this means is some people that weren't classified as severe were still hospitalised, because that's obviously smaller than that. And deaths out of this 1.2 million, out of the official documented infections, deaths were 41. So we can see we've got a lot of data here to sort of get good results, get good information from. Now, the results. Vaccine effectiveness at 14 days through to 20 days. So they looked at how effective the vaccine was in this time period, 14 to 20 days. Now, in the light of subsequent data, we know this is not enough. We now know that the full immunity effect of the Pfizer vaccine takes a good 3 weeks or more to kick in. So people that were only followed up for 14 days, they wouldn't be enjoying very high levels of immunity yet. People that have been vaccinated for 20 days would be enjoying a higher level than the people that had only been vaccinated for 14 days, but it was still building up. But nevertheless, this is the data we've got. So we're comparing people that were vaccinated between 14 and 20 days and the vaccine effectiveness at 7 or more days after the second dose. So what was happening in Israel is on day 1, or day 0 really, people were given the first dose. On day 21, they were given the booster dose. So they were looking at what happened in days 14 to day 20 and what happened 7 days after. So that's 28 days after the first dose, a minimum of 7 days after the second dose, and they were given the second dose then. So we've got lots of information from here and from here. Now, what does this mean for the UK, for example, with a one-dose strategy? Well, it's actually quite limited. Everything up to 21 days when they had the second dose is going to be completely valid, of course. But if they're taking data 7 days after the second vaccine, can you see that's also 7 days after the second vaccine? It's also 28 days after the first vaccine. And we know that after one dose of the vaccine, the immunity builds up strongly from day 21 to day 28. So for all we know, the second vaccine wasn't actually helping the immunity. It was actually because it was longer from the first vaccine dose. The immune system had had more time to generate an immune response. Now, having said that, the second dose would have some effect, of course, but it does mean that we can't get too much information about the UK situation. Although there are reasons to believe that the UK is on the right strategy, and we still firmly believe that they are. But to be completely scientific about this is only giving us information about the second dose if it's after 21 days. Right, so let's have a look at it anyway. So obviously this is compared to the unvaccinated group, because we have to have the unvaccinated group to compare it to. Now I'll give you some numbers and then I'll give you some graphics, and between the numbers and the graphics I really believe this will make sense. Because there's no substitute for going back to the original papers, getting the original data. So people that had the vaccine effectiveness after between 14 and 21 days in documented infections was 46%. 7 days after the second injection, at least 7 days after the second injection, it went up to 92%. Now, all of that is not caused by the second injection. That massive increase is not caused by the second injection. We now know that a large part of that increase from 46% to 92% efficacy against documented infection is due to the fact that it's simply been longer since the first dose was actually given, because we know that the immunity builds up from day 21 to day 28 even further. But this doesn't differentiate between those. But that's good, and of course that's compared to the controls who hadn't been vaccinated. One of the problems was actually in this study that as the study went on, more and more people were vaccinated, and of course people didn't want to turn the vaccine down just because they're in the study. So as people were vaccinated, what they had to do was actually take them out of the control group. And in some cases, they actually put them from the control group into the vaccination group and just recruited someone else to be in the control group who hadn't yet been vaccinated. But of course, that meant the people in the control group kept getting younger, which means these are really minimum figures of efficacy. So that was documented infection. Symptomatic infection. So symptomatic infection, 57% efficacy between day 14 and day 20 after the first injection, going up to 94% efficacy at least 7 days after the second injection. But again, it's been more time since the first injection. But these are just brilliant numbers. Hospitalizations, 74% protection compared to unvaccinated people in 14 to 20 day time period after the first injection. 7 days after the second injection, which is 28 days after the first injection, 87% less people were hospitalised. But the last hospitalisation in the vaccination group was at 32 days. So in the people that were vaccinated, none of them were vaccinated after 32 days. That's 32 days after the first dose on day 0. So the lesson here really is very potent. This is taking like a full month to build up these high levels of protective immunity against hospitalisation. What about severe disease? Well, 62% within 14 to 20 days after the first injection, 92% 7 days after the second injection, 28 days after the first injection. Again, very good efficacy figures. But we can't break down how much of that protection against severe disease was caused by the second injection, how much of it was just caused by waiting for longer after the first injection. But the last severe case in the vaccinated group was 30 days after the first vaccine. So there was no severe cases in the vaccinated group 30 days after the first injection. And effectiveness in preventing death, 72% for 14 days through to 28 days after the first dose. So 72% protection after one dose from dying. Good reason to believe that after another week or two that would be even higher, even without the second dose. But deaths at 44 days into this study, the vaccinated group, there had 9 deaths, the unvaccinated group had 32 deaths. That's where we get that 72% efficacy from. But the last death in the vaccinated group occurred at 30 days after the first injection. After that, there was no deaths in the vaccine group. So we see it is taking this month to kick in. Just frustrating. It would, ideally, from our point of view in the UK, that if they'd left all the vaccinations for another couple of weeks, we would have had much more useful data for the UK. But let's look at these on graphs. So what we have here is that the red line is the non-vaccinated group and the blue line is the vaccinated group. So these were documented infections. So we see that for the first, 14 days after the injection, we don't really see a protective effect at all. But then people who weren't vaccinated went on getting more and more infections. People who were vaccinated, it started tailing off and became quite a bit less further on. Now, that's just documented infections. This was symptomatic infections. And again, we see no difference to begin with, but then we see this unvaccinated group getting more symptomatic infections. These are cumulative numbers on this sort of graph. But here we notice that in the vaccinated group, after day, after day about what's that, 30, 38 or something there? So 35, 36, 37, 38, 39. It looks like after day 39 or 40, there was no increase in cases. There was no new symptomatic cases in the vaccinated group. Whereas in the unvaccinated group, it did go up. Hospitalizations. Now, it's good to see that even in the unvaccinated group, these did level off because the number of cases in the country was going down. But they levelled off much earlier in the vaccinated group. So what we can say here is from day 28, 29, 30, 31, 32. From day 32 in the vaccinated group, there were no hospitalisations, like none, in the people that have been hospitalised. As I said, how much of that was due to the initial dose of the vaccine at day 0, how much that was boosted at day 21 when the Israelis give the booster dose, we don't know, but pretty impressive figures there all the same. And we can't say from that we would expect similar figures in the UK because we're not giving the second dose at 21 days. But what we can say is the immunity effect does take longer than the 21 days to reach its full efficacy, as we're only giving one dose in the UK. Severe COVID-19, again, vaccinated group much lower from day 7, day 14. The gap becomes bigger as it goes along. And then in the unvaccinated group, it did level off there. But in the vaccinated group, it levelled off much earlier. So we can say that there was no severe cases of COVID in the vaccinated group. And remember, this is 500,000 of them after day 30. So what we're saying there is in 500,000 people that were vaccinated, after day 30, after 2 doses, no one got severe COVID. I mean, this is just so impressive, this protection against severe disease, albeit this being after the second dose. And it won't tell us what we were craving to know for the English data, but it simply won't tell us. It's not there. This is deaths. So again, in the vaccinated group, no deaths after the 30th day after the first dose, which is 9 days after the second dose. To what degree this effect will be facilitated with one dose, of course, we can't fully know. But this is impressive data for the efficacy of the Pfizer vaccine. Very, very impressive indeed. Now, so deaths, last death in the vaccinated group, I've got it as day 30 there. That's about what we said. Right, conclusions. Consistent with a gradual daily increase in vaccine efficacy. This is taking 3 to 4 weeks to 5 weeks to even longer to give us maximum vaccine efficacy. And as we saw, the gap between the unvaccinated group and the vaccinated group gets bigger as you go along. Not much difference in the early stages. This is a nationwide mass vaccination setting, so that the Pfizer vaccine, mRNA vaccine, is effective for a wide range of COVID-19 related outcomes, as we've seen. This is a direct quote from the paper. Consistent with randomised trials. So that 28,000 people from the randomised trials did give us pretty good data. Now, I think that as far as this goes, those graphs are still supporting the UK strategy, given that we can't vaccinate everyone as quickly as we would like. I think this strategy is still supported by that. And take this in context with the Scottish data, and I especially think it's still supported. Now, let me just give you a bit of a reason why I'm thinking that. So we know that when a vaccine or a virus enters the body, that is described as an antigen. An antigen being anything the body recognises as being foreign. And that will cause the body to produce the immune response. And the immune response is the antibody. But there's also many other parts of the immune response as well, as we'll see. And the antibodies, their proper name is immunoglobulins. That's their proper name. They are immune proteins, immune globulins. Now, when the antigen first comes in, the first antibody that is produced pretty quickly. That's called an immunoglobulin IgA M, an immunoglobulin type M. Now this is actually like a pentamer. It's got a middle bit there, and it's got 1, 2, 3, 4, 5 bits coming out of it, with a Y shaped on the end of each. And these are the actual arms. So that's the initial antibody that's produced, the immunoglobulin type M. And you might remember that we said antibodies are like, where have I gone? Antibodies are like our bodies, and they have 2 arms. So I can grab that, I can grab one virus with that, I can grab one virus with that and start destroying them, as I can grab more than one at a time, and I can agglutinate them all together and neutralize them with my two active arms. But the actual hand, the actual, it's called the multivariable region on the end of the antibody in the IgMs is not as specific. In other words, it's not quite as good at gripping things. It can grip it, but not very well. But over time, the next antibody is produced, and that next one produced is the IgG. Immunoglobulin type G is the next one produced. These don't last for as long, so these are the initial fairly non-specific response. But then after a bit of time, we get the IgG produced, and this is more specific. So the immunoglobulin type G, and that's just a simple antibody, just that Y-shaped molecule. The body here with the 2 arms here and here. But these arms are more specific. In other words, on the IgGs, so whereas the multi-regional parts on the IgMs aren't very good at gripping things, they're a bit crude. They're not a very good fit. They're not a very good grip. With the IgG, it's a much more specific grip. And with the IgG antibody, when the antibody grips the antigen, the specific part of the antigen that it grips is called the epitope. Then it grips it well, and it makes a good job of it. It becomes much more specific. So they're made first, and then they're made. But the other thing that there is that there's immunoglobulin type A's, which they're this shape, IgA, the immunoglobulin type A's. And these are found mostly in the mucus, protecting the lungs, for example. And, you just don't seem to read any data about immunoglobulin A production after vaccine or SARS coronavirus 2 infection. So very limited amount of information on that, unfortunately. But it's reasonable to imagine that these IgAs are floating around in the mucus in the nose and in the airway, protecting us against the infection, as well as specific immunoglobulin type A's are generated in response to the antigenic challenge of the virus or the vaccine. So that's going on. Now, the next thing that probably happens is the production of the NK cells. Now, the NK cells are the large lymphocytes. So they're like the B and T cells, but these are larger versions of it, and these will directly kill. These are cytotoxic cells. They will directly kill virally infected cells. Now, these used to be considered as part of the innate immune response. We now know it's part of the specific immune response as well, but they are probably taking a bit more time. So you see what I mean? First of all, we're going to have the IgMs, then we'll have the IgGs, then the natural killer cells will probably come along at some stage. So all of these things are taking time. But then the next part of the response is taking even more time. Of course, all these immunoglobulins here are made by the B cells, the beta cells. And as we've said, initially, they're going to produce non-specific IgMs. Then they'll start producing specific IgGs. But to make it more specific takes time. This all takes time. And then, perhaps in most cases after that, it's the T cells are going to come along. Now, the T cells, again, they're another form of small lymphocytes, and there's 3 sorts. There's helper. You might have heard of T helper cells. It's the T helper cells that are infected in HIV. So in HIV, the T helper cells aren't there to stimulate the B cells to produce the antibodies, and that's big part of what causes the immunocompromise in HIV. So there's the T helpers, then there's the T cytotoxic. And cytotoxic means they kill cells, and there's also suppressor cells which regulate the whole immune response. And of course, these are very important as well in COVID-19 because they're going to limit the inflammatory response. But it's these T cytotoxic cells that are probably one of the key players in immunity in this form of viral infection. Now, the T cytotoxic cells, what they will do is once they are produced, and it takes a bit of time to produce them, but if there's a cell here that's virally infected, so suppose there's a virally infected cell here, and the SARS coronavirus 2 virus is in that cell, then what the cell manages to do is it can manage to smuggle parts of the viral protein onto the surface of itself, onto the surface of the body cell. And of course, this will alert the body to the fact there's an infection there. And then these cytotoxic lymphocytes will recognize that this cell is virally infected. They will recognize this part of the viral protein that's been ejected from the cell by the cell itself. And this cytotoxic lymphocyte will then release chemicals, which basically kills this whole cell. Kills the whole cell. Now, it's bad because it kills the body cell, but it's good because it kills all the viral particles inside it. And that is probably the main response that's protecting us against severe disease, hospitalisation and death, but it's taking time. So what I'm saying is, so if it's 3-4 weeks after the first dose that this T cell response becomes significant, then it could well be that these excellent results in Israel that we're seeing 4 weeks after the first injection are at least partly the result of waiting 4 weeks. Now, of course, we need to give the second booster dose at some point, but the data doesn't allow us to differentiate which is which, and part of it might simply be that it's more time, because we know it's going to take 3 or 4 weeks to build up a full response. So it could well be that this T cell response turns out to be the main thing, this T cytotoxic response. Then, of course, the B and the T cells are also going to make memory cells. So the vaccine produces memory cells. Well, the infection will produce memory cells, but the vaccine produces memory cells. And these are going to hang around. We don't know for how long. We're hoping for many years. We hope even for a decade or two. We simply don't know yet. But these are going to hang around so these can be activated and differentiated into active B and T cells really quickly, including they can rapidly differentiate into cytotoxic T cells to destroy virally infected cells. And again, this is going to take time. So this is why it's so important that the whole response is taking time. That's why in the initial Israeli data there was more cases in the first 8 days after vaccination, because it altered people's behaviour. They didn't realise they had to wait for 3 or 4 weeks. But of course, from the Israeli data, we can't differentiate between how much of the beneficial effect we've seen is caused by the second dose and how much of the beneficial effect is simply caused by waiting for 28 days after the first dose of the vaccine. It won't tell us that. The data simply won't tell us that. But that's kind of a bit of intriguing physiology that might it will be influencing this to some extent. The nature of the immune response, and the fact that that happens first, then that happens, then probably that happens, and then that happens, and then that happens. It's like a sequence. It doesn't all happen at once, and it takes time. So interesting things. I mean, let's give the takeaway message here. Any vaccine is massively better than none. And we know that all of the vaccines are preventing against severe disease, hospitalizations, and therefore deaths. So hope that was interesting on the on Israeli data. More to come on the Israeli data, I'm sure. But what we're really waiting for from the UK now is the data on the Oxford AstraZeneca vaccine, which really should be out within the next week or so, hopefully. Lots of Pfizer data coming through because that was the first. Of course, the real world data on the Oxford AstraZeneca vaccine protecting from hospitalisations in Scotland was already there. It was 94%, which was somewhat higher than the Pfizer vaccine. So lots more exciting data to come, but so far it's looking really better than we could have hoped. Really better than we could have hoped for good vaccination data and efficacy. I think that's all I wanted to say, really. So thank you for watching.